Welcome everyone to our webinar on Blueprint Neurotherapeutics Network for Biologics with the acronym BPN-Biologics. My name is Anne-Marie Broom. I am a program director in the Division of Translational Research in the Institute of NINDS. And hosting the presentation for today. So welcome everyone. We'll go through first some logistics associated with our webinar activities today. So this webinar will be recorded. The recording will be made publicly available on our website. PDF copies of the slides with embedded links will also be emailed to registered attendees. Please use the Q&A box that you see in your Zoom dropdown to post all questions. My Vanna White, Jules Bachman will be fielding those questions and will answer them during the question and answer period at the end, as well as direct answers that may occur online for that. Project-specific questions, however, should be best addressed by sending us your specific aim pages, and we can set up a call to address those specific issues that are not answered within our webinar today. Okay, so let's get started. So NIH had developed BPN-Biologics as an expansion on an already successful Blueprint Neurotherapeutics Network program. And this actually came about because the goals of combining strengths of NIH and industry expertise for neuroscience, for drug neuroscience discovery, was clearly a gap within the field. And so what it actually allowed us to do is to have NIH investigator-initiated ideas that have a solid scientific premise with experts in disease biology specifically associated with their assays and models. And add to that industry expertise and availability such that consultants with extensive pharma experience across all the R&D disciplines would be able to interact with our NIH investigators and utilize pre-established industry standard contract services. The end goal then is to actually de-risk projects in advance and advance those projects to the clinic and then hand them off to commercial or pharmaceutical investment success. And one of the exciting parts of this is also that the investigators and institutes that are requesting funding would maintain their intellectual property and license and drive it forward as well. So looking at our program goals, in our BPN-Biologics program broadly accounts for the different biologic modalities, including large biomacromolecules, cell therapeutics, as well as gene therapeutics, both oligo and viral-based biologics, as in addition to disease indications and entry points. We provide non-dilutive grant funding and necessary resources through NIH established contracts and consultants, small matter experts that are typically lacking in our research community. And what this really makes available to the investigator is de-risk potential therapeutics to the point that industry will invest in them and allow potential new drugs to reach the patients efficiently. So what we really want to do is identify the best ideas that will translate seamlessly in the NIH research community through this funding opportunity and infrastructure, with an eye on the fact that we are preserving the PI and institutions' intellectual property to facilitate licensing of those products. So our funding opportunities that have been published, if you search for them in Google, are for both academics and small business. The links will be available in the slides. There's a UG3, UH3 cooperative mechanism for all applicants, as well as a U44 fast track for SBIR eligible small businesses. Grant funding to support these biotherapeutic and development projects into the clinic is the ultimate goal of the funding. And really, it gives you an opportunity to access NIH-funded consultants that run the gambit of drug discovery, subject matter, as well as contracted, vetted research organizations to conduct a variety of different levels of experimentation. In addition, it puts you in a relationship with expert NIH project managers and science officers to help guide and shepherd you through this whole process. And so our upcoming receipt dates for this, the first one is August the 10th, 2021, with the second one being February the 9th, 2022. And those will be reviewed by a multidisciplinary special emphasis panel afterwards. So the BPN is a trans NIH program with 9 participating IC within. And on this slide, you actually see the program directors that are available for contact within the various ICs. And this will also be submitted as link outs in the information in your PDFs following this presentation. Now, I don't expect that you'll actually read through all of these or catch everything for these, but suffice it to say, it is very important that as general information, we want to make sure that your project focuses on a single indication that falls within the mission of one of the participating ICs. And so what we really recommend that you do well in advance of your submission of your application is to speak with your IC reps to make sure that it is within mission and to also get any added assistance during that process. So this breaks down more in depth the actual customized combination of infrastructure, expertise, and funds that you can expect through BPN-Biologics. As a project PI, you will receive grant funding for biologic activities, free access to our network of CROs, and a team of consultants and NIH staff who will manage the project. And for each project funded under the FOA, you'll actually set up a customized lead development team, which we refer to as an LDT. And that LDT will be co-chaired by the PI/PD of the project, as well as BPN Biologics consultants, and will include members of the PD/PI's team and additional BPN Biologics consultants and NIH staff. And so that helps the process. The LDT actually develops an overall strategy for the project, including milestone-driven proposals, planned studies to be conducted by the BPN Biologics contractors, and coordinate activities across these different research sites. And the bullet points down below here really strike home what those value-added propositions are, in that we provide these contract resources that are tailor-made to support each project. We have a flexibility of how you use both the contracts and your PI in-lab experimentation from the grant support, and still PI teams' intellectual property is retained by the PI's institution. Now, this brings up the concept, as pointed out before. Your PI institution will be responsible for the intellectual property. So inventorship is determined per US patent law, and NIH blueprint has no stake in the IP. And what that really means is that prior to the grant award, the PI's institution must have upfront IP or licensing agreements in place with all potential inventors and must demonstrate this within the application. These agreements must address two principal points. Who will hold title to the IP on the new biologic or therapeutic, as well as what the royalty arrangements are. And so you're going to want to specify this within your application. IP agreement should aim for unencumbered IP, so there should be no legal hindrance to your being able to drive this forward into the commercial realm. And it should be consistent with blueprint program goals to create licensable products, right? The caveat here is also that your PI institution will be responsible for assembling and submitting the IND application and scheduling regulatory meetings with the FDA. The LDT that I mentioned in the last slide will be a participant on all those FDA meetings and help tailor forward progress based on those recommendations from the FDA. So make sure to include support for this activity within your application plan. NIH staff and consultants on the LDT must be included in all of those meetings, and it must spell that out within the application. So let's look at the actual structure of the BPN-Biologics program. Projects can enter into either the discovery or the development stage of the program. The discovery stage is defined and used for lead characterization and optimization to improve the potency and or suitability for ultimate clinical testing. The development stage is a developmental stage that is utilized to advance a developmental candidate through IND enabling toxicology and safety studies and ultimately into phase 1 clinical testing. And as you see here, it demonstrates a major point in that all projects begin with a UG3 phase. Now, if you are applying to an SBIR component of this, the U44, then realize that instead of UG3, UH3 language, it is the phase 1 and phase 2 language instead with that. Make sure that you realize that all projects should reach the clinical trial stage regardless of whether they enter at the discovery or developmental stage within a maximum of 5 years. And so how you navigate that in your application is up to you, but keep this maximum number of years in mind. So looking at our application guidelines, the UG3-UH3 is a research project cooperative agreement with a length of time of up to 5 years. The UG3 component of this in either the optimization or preparatory for the development is up to 2 years. The budget is not limited, but must reflect actual needs of the proposed project. The U44 is a small business innovation cooperative agreement, subset under the fast track. The phase 1 can take up to 2 years, and the phase 3 can take up to 3 years. There are caps for the budget in the U44, and that the phase 1 can have budgets up to $700,000 per year, and the phase 2 can have up to $1.5 million per year, but must be reasonable and appropriate for this. Also, recognizing that in the budget, if you go over $500,000, you do have to have an additional step in which you will submit to the appropriate review. So when looking at a phased grant mechanism, realistically, what you're doing is after you're awarded the grant, the LDT will help you establish clear, quantitative, and definitive milestones that are essential for the success of moving your product from start to finish within the program. There are annual go, no-go milestones that are created with you and the LDT that must be accomplished at the end of each year. The transition between a UG3 to UH3 occurs via administrative review and successful completion of these go, no-go milestones for the phased mechanism. So let's look at the particulars of the FOAs. So if you come in at the discovery stage, there are relatively clear entry criteria in which we want to make sure that we have written it broadly to include everything, but that the modality and the disease indication are going to determine the trajectories and the realities that you need to address for each of your modalities and success within the program. And so please remember that you have only 2 years of optimization activities. So to enter into the discovery stage, what you want is identify one or more lead biologic agents that have been sufficiently profiled so that the parameters that still need to be optimized can be quantitatively specified. You want to establish preliminary in vivo efficacy, and there's a little appendix stars under proposals may be considered without in vivo ex vivo efficacy, but there has to be a clear rationale backed up with compelling in vitro data to account for this exception. And target engagement data using agents in relevant animal models specific for your disease. Key in vitro and in vivo assays are suitable to drive characterization and optimization of the biologic agents. And there should also be, as I mentioned in the prior slides, no obvious legal or intellectual property constraints. The end result of the UG3 mechanism is that you have a clinical candidate in hand with appropriate bioactivity, stability, manufacturability, and bioavailability by the intended or route of administration for development, further development within the developmental phase. Target engagement with defined minimal and optimal doses by the intended route of administration and in vivo efficacy data when applicable for this. Oh, sorry. So let's look at some of the examples of projects that meet this goal that can be done within the discovery UG3 phase. And that includes characterization of identity and properties, lead optimization of characteristics to improve effectiveness, diminish toxicities, improve ADME results, optimization and or qualification of appropriate assays for pharmacokinetics, a target engagement, biodistribution, determining the optimal route of administration is example of an activity that can be supported under the discovery phase, and demonstrating adequate stage appropriate preliminary safety tox, dose ranging or dose range finding toxicology studies. Then once you have met this goal, you will enter into the developmental phase, the UH3 phase, beginning with preparatory activities listed below. So all applications proposing to enter into the development stage will begin with a UG3 funded stage for this of up to 2 years. The entry criteria for the development stage include a strong body of data linking the putative therapeutic target to the proposed disease indication. You want to rationally lay out the biological activity by the planned route of administration with exposure levels for activity being achievable based on ADME properties appropriate for the intended clinical use. And you want to demonstrate that your institution has the ability to develop or commercialize a proposed biologic and drive it into phase 1 clinical trial. Because the ultimate goal really is to submit an IND package, have it successfully activated so that you can start a phase 1 clinical trial. Excuse me. So, when looking at what constitutes development stage activities, you can have development stage activities in preparation for the IND enabling studies that include non-GLP toxicology studies, formulation experimentation, scale up manufacturing and stability studies, repeat of in vivo studies in the same or different animal models, right? A one-shot repeat, a pre-IND meeting with the FDA if that has not already been conducted for this. Now, the milestone checkpoints to start your IND enabling work covered within the development stage are DRF tox studies, both rodent and non-rodent, that have shown an acceptable safety margin. Manufacturing related development processes have been established, and that you have an optimal delivery route determined, and it is feasible for the proposed indication and patient population. So then that drives you into the development activities, which can include the following, which are cGMP manufacturing of the biologics for IND enabling and or phase 1 clinical testing, IND enabling safety tox studies to determine the basis for the clinical dose extrapolation using your relevant animal model, and preparation and filing of the IND document, as well as phase 1 clinical trial plans. Now, what should go into your clinical trial design? Examples of clinical trials which would be supported under our BPN-Biologic FOAs would be signal dose or SAD, which may be placebo-controlled or open-label studies. Multi-ascending dose studies, which may be requested only if an agent has a short half-life, as well as clinical trial outcomes that may include safety, tolerability, PK/PD, target engagement, and target modulation endpoints for this. BPN biologic contractors can conduct phase 1 clinical trials for investigational biologic agents that may include large biomacromolecules in healthy volunteers or demographic subsets of healthy volunteers. If your clinical trial plans to utilize only diseased patients and enroll them, you're going to need to supply your own clinical site and propose a plan that effectively details the information of that plan. Applicants are strongly advised, I can't emphasize that enough, to discuss your plans with NIH program staff prior to submitting your application to determine whether a clinical trial is feasible within the proposed timeframe and or available BPN biologic contract resources are available. Okay. Now, some general tips in preparing for your application. First and foremost, it is very important that you read the funding opportunity announcement carefully. Many questions can be answered by careful perusal of the information provided within the FOA. Also, discuss your proposal with NIH BPN Biologics Program Director, that's me, or our staff, as well as the PD of the home institutes for disease interest of your proposal. You can also contact SBIR program directors for SBIR specific issues. And when preparing your application, you want to make sure to put forth a solid scientific preliminary data packet to support your proposal and address the rigor of that data in your research strategy section. You want to Think carefully and address obvious criticisms of the biology. The more clear and plain spoken you can make your application on the front end, the easier the process will be for review of your application and subsequent downstream funding. Clearly indicate what will be done as part of the grant and what is expected to be done by the BPN-Biologics contractors. This actually occurs on a spectrum. It can have the PI do all the research experimentation within their own laboratories, or they could have all BPN-Biologics contractors do the work. However, it is more likely that some mixed PI laboratory and BPN-Biologics contractors will mingle and come up with the two and not be on that either extreme. The last thing is to also include budget pages for all years of the grant, not just the overall and not just for the first one. So budget pages need to be included for all years. Another very, very important aspect of your application is that it addresses rigor. And so in the preliminary and supporting data section, explicitly discuss the quality of the data presented in prior publications in a very detailed manner. Some questions you can utilize to ask yourself whether you are sufficiently answering this are were they done in a rigorous manner, utilizing randomization, blinding, inclusion, exclusion criteria, and the appropriate power analysis. And so address that within the preliminary and supporting data you provide. Within the approach, provide details for your controls being used for each type of experiment and appropriately highlight potential confounds like surgery exposure, genotyping, variability, and human placebo effects. Make sure to include details within the experimental design about the reduction of potential bias, including blinding randomization and inclusion-exclusion criteria for that. Make sure that you describe the source of the data on which the sample size estimation or quote unquote power analysis is based, and detail that within the analysis itself. So things to do in preparation. Make sure that you complete your required registrations at least 6 to 8 weeks in advance of the receipt dates. If you are applying to the UG3, UH3 FOA and any proposed budget year that exceeds $500,000 in direct costs, excluding the F&A costs, then you need permission to submit. That actually is a red flag, which means that you must request 6 to 8 weeks prior to the submission date. Make these requests prior to that. Talk with your tech transfer and business development group, planning in advance for funding, patents, and licensing activities that will come out of your biologic development and commercialization. Now, this is also a very important slide because it actually outlines things that you should not do. So do not plan for this to be the sole funding for your laboratory. Milestone-driven programs can end abruptly based on their aggressive pipeline through the process. Do not under-resource your budget to avoid limits, right? Really ask for what you need and justify that it is reasonable and within expectation. Do not plan for BPN-Biologics contracts for disease biology or one-off in vitro ADMET experiments. Do not code your application as a clinical trial if your plan is to use a BPN-Biologics contractor, right? So make sure that you check the box that says that it is not a clinical trial. If you do plan to run your own clinical trial, then you will indicate clinical trial, and it will be considered a delayed onset clinical trial. And that's what you will be describing. More information can be received through the following website. So we put the link in there under BPN-Biologics. You see our face page for this when you click on our link. And if you notice along the side in our child pages, there is a significant number of important and interesting information that I recommend that you examine in addition to reading your FOA, such as how BPN biologics work and the virtual pharma model, what resources are available, as well as the important frequently asked questions. Oftentimes, your question is similar or along the same vein, and you can easily find some answers on our website. Now, if you have questions after our webinar is concluded, additional information can be found on our BPN Biologics website. For project-specific questions, please contact either myself at the email provided or Jules Bachman, our health program scientist, as well. And I'll actually stop here and open up the field to questions associated with BPN Biologics.
Okay, great. Anne-Marie, can you hear me okay?
I certainly can.
All right, so we're going to start with a couple questions that we received prior to today's webinar. The first is regarding entry criteria. Could you please detail what exactly qualifies as established preliminary in vivo efficacy data and target engagement? And sort of a follow-up to that is, if you have preliminary data that's not yet published in peer-reviewed journals that's included in the proposal, would that be considered too premature or non-competitive for these FOAs?
So, so very, very good question. So in essence, effective and established preliminary data is such that you have strong rationale for what you're proposing and that it is viable and will be successful using in vivo efficacy data, biodistribution, target binding assays, downstream endpoint analysis for that, right? It is also appropriate to utilize prior publications from your laboratory to support your preliminary data coming through this PAR. Now, the second question was, if the preliminary data has not been published, could it also be appropriate and included in abstracts and everything? Would this be considered too premature? The answer is no. However, you want to have substantive data to back that up. And oftentimes we recognize that the preliminary data may not be published because of intellectual property rights that are occurring for this. So you want to make a strong and rational case for the likelihood, as well as back up the current field of knowledge to support your preliminary data. Okay.
Next question. Since NIH consultants and CROs are expected to partner with the PIs to design and carry out the relevant IND-enabling studies, how much detail should the applicant try to provide in the proposal about study design, milestones, and other aspects of biologics development?
All right, so what we would request is that for the PI planning to use NIH contract resources, your general design, expectation, and outcomes should be proposed and spelled out well. Generally speaking, when referring to the milestones, you want to have a general anticipation of what appropriate go, no-go advancement criteria may be to declare the development candidate ready for the next stage, right? More detail, however, is requested if you are planning to do the experimentations in-house, right? So provide sufficiently detailed information for work that will be done within the PI's laboratory. Did I get all of the, was there a second part?
No.
No,
but
okay.
one more question
All
for
right.
this batch. Should the applicant try to contact appropriate individuals on the BPN resources list to solicit their input in preparing the proposal?
So that answer is no. It is not required or necessary to contact the specific BPN resources. They're vetted through NIH and INDS to be able to conduct the experiments. However, I would strongly urge that you have these types of conversations with relevant pharma and industry companies to assist you in recognizing what these planned experiments could be to help fill in and determine your gap assessment for experimentation.
Can STTR respond to PAR 21-233?
STTR? No, this is
STTR.
Yes, so no, this is SBIR phase 1, phase 2 fast track.
Mm-hmm. So I would still encourage you to contact us and we can put you in touch with our small business group who can help determine, if you're really eligible for SBIR versus STTR and how we might be able to make that work. But you should still be able to apply to the UG3, UH3. Okay. Are there any restrictions on the disease types, i.e. neuropathic in nature, to be eligible for BPN, particularly meeting NINDS mission?
So our goal is neurologic based and neuromuscular based. For that, I would recommend discussing within our NINDS disease expert PDs to assist in determining mission fit for those, as well as the other ICs if you think that it will come through those ICs as well.
And just to add on to that, within NINDS mission, we do not have priorities for particular indications or disorders, but other ICs may have priorities within their mission. So that's another reason to contact us early on.
Good point. Thanks, Jules. That's excellent.
Okay. Next question. Does this program fund peptide drug development?
Yes. So peptides falls under biomacromolecules. Depending on the size of the peptide will determine whether it's BPN-Biologics or BPN small molecules, just on size. But in general, the answer would be yes.
Okay, I'm going to paraphrase the next one a little bit. They're talking about starting a new clinical trial for presumably a second indication. So they have a clinical trial started in cancer pain, but those patients are quite complicated due to variations in cancer. This is a pain therapeutic and could be more specific applicable issues. So I'm saying the question is sort of, can you come in for a second indication in BPN-Biologics if you're already at a clinical trial stage?
Coming in for a second indication? I don't believe so. Jules?
If your biologic is already a candidate in first-in-human clinical trial, then you are beyond the scope of BPN-Biologics. But if there's additional preclinical, non-clinical activities that you would need to do for this second indication, then I think that you should contact us and to have a discussion. Particularly if this is a pain therapeutic, there are a number of funding opportunities open through the HEAL initiative. So please reach out
Yes.
to us or other program directors in the HEAL initiative that can direct you to appropriate funding opportunities at any stage along the therapy development process. Okay, we have a pending STTR submitted in April. Can we still respond to the August receipt date? So I can just jump in on that one.
Yeah,
That
yeah.
depends on, probably not. So you need to have received your summary statement in order to submit a new application that has any significant overlap. Okay, SBIR U44, the budget limits, does this still require permission over 500K?
So for the under the SBIR?
That's the question, yeah.
Yeah. So this is for the UG3 UH3, yes, it does. For the U44, Jules, you'll have to back me up here. Does it also still require an ARA?
No.
No. Okay. Yeah, because their SBIR budget limits are different than the UG3 UH3.
Mm-hmm. But it's important to note that for the UG3 UH3, the 500K is in DC without consortial arrangements, whereas for the SBIR, the guidelines that were proposed are for total cost.
Yes. Yes.
Okay. We already have an SBIR for initial preclinical development of the biologic. Can we still apply for the discovery program?
So this goes back to what Jules was referring to in the past. So there would be, I'm assuming there would be overlap with the outcome of this and wouldn't be applicable for the discovery program, right? Because you're in that non-clinical development of the biologic. But we could review your proposed specific aim page more clearly after the fact, if you'd like.
Yeah, so I guess it depends on the activities that you're doing for your current SBIR. If you have a phase 1 SBIR that's really early on, and everything that you'd be proposing in the discovery BPN-Biologics application would be after that, then that's still applicable. So please reach out to us with your specific aim. Okay, how much preliminary data will be required for the UG3?
Okay, so what you're actually trying to do here is to provide effective proof of concept data for this, which is variable based on the type of preliminary data you're providing. In essence, you want to prove the goal of your proposed project and demonstrate that, argue definitively that it's viable and feasible and will be successful to have that downstream effect during the development process.
Can a CRO in Europe be proposed?
The CROs that are NIH, if a PI requests a consultant research organization for their own partnership, yes, you can, but it has to be specified within the for that activity.
Does this program fund biologics development for rare genetic diseases?
Yes. And we also have other up and coming ultra rare genetic disease gene therapies available as well. So stay tuned.
COVID-19 post-acute sequelae include a variety of neurological conditions. Are studies of interventions for these conditions covered by the Neurotherapeutics RFA, and if so, by which IC?
Off the top of your head, do you know, Jules, which IC?
I do not. So this is new and developing, and we also have COVID-19 specific RFAs that might be more appropriate depending on what you want to do. So I think this is an example of please contact us and we'll follow up. Okay. We have an institute and company entities. Which PAR is more appropriate?
So the UG3-UH3 is proposed by the PI as opposed to the U44, which is proposed by the small business. So depending on the goal of your proposal will determine which route you take, the UG3 or the UG3-UH3 or the U44. We can help with some of that assessment, but really that's a decision that needs to be made between you and your small business entity.
Mm-hmm. So the assumption here is that your company is SBIR eligible, or at least that's the assumption that I'm going to make. So the U44, which is the SBIR version, does use SBIR set aside dollars, and then you have to follow the SBIR rules based on how much work is done in-house versus outside. So this also might be an example where you can contact us and we can loop in our small business associates to help make that decision in terms of how those rules might guide you. But it ultimately would be up to you.
Right, right.
Okay, let's see, returning to the previous STTR question, section 3 seems to suggest that STTR is eligible, no? So STTR and SBIR are technically two separate set-asides from Congress. So they have set-aside funding opportunities. So a funding opportunity is either for STTR or SBIR. But if you have additional questions on eligibility, please contact us and we can work this out. Okay, can you apply if you have an R61, R33 pending on the same biologic? So if pending as in it has not been reviewed yet, then no, you need to wait till you have your summary statement to submit another application that has overlapping aims. Maybe already answered. If I currently have a phase 1 STTR grant, can it switch to development phase, i.e. equivalent to a phase 2? So that one is also something I think that you should contact us to loop in the small business. The U44 is a fast track, so you could potentially apply to the development stage of the FOA, but you would still need to apply as a fast track application. So you would have both a phase 1 and a phase 2.
Yes.
I believe that is all the questions that we have.
Very good.
All right. If anyone else has questions, please. Another question came. Can you apply if you already have an R61, R33 phase for the same biologic? If it's the same biologic that you are developing further, then yes. But if the activities that you are being funded for, you're already being funded for them now, then you cannot propose to do them again in a new application.
Yes.
But if you're referring to Ignite, then that is meant to be a feeder program into BPN-Biologics. So it's meant to get you to the entry criteria for this program.
Yes.
Okay, oh, bunch coming in now. I missed this earlier. Does this cover gene therapy products?
Yes, it does.
Okay. How does this mechanism relate to the R61, R33 mechanism?
I think this is what you were outlining, Jules, when you were saying that if it falls under the Ignite program, it is our feeder mechanism.
Yeah, so I mean, sorry to interrupt.
No, go
I mean,
ahead.
if you're referring to the Ignite, like there are other R61, R33 programs in other ICs as well. So I don't want to assume that they're referring to Ignite. But in general, in R61, R33, it's similar in that there are two phases. There's usually an administrative review based on project milestones to determine if the R33 is awarded. Whereas these are cooperative agreements, so there's much more interaction and involvement by NIH program staff. Information on the BPN contractors. Where can we find pertinent information for what activities for which contractors?
So on our website, we actually have started to provide information for the resources of the CROs for this. So you can certainly start looking there. For instance, it covers PK tox studies and safety studies, as well as modification, chemistry modification, and some clinical trials activities for that. Manufacturing, stay tuned, it will be added shortly.
Okay. Are there CROs who can perform non-human primate studies? If so, how can one estimate these costs in the budget? Are the studies performed by CROs part of the application budget?
So it's broken down into two questions. NIH CROs can perform HP studies, but please do contact us to discuss these particular studies. If you are asking whether the CROs that you have relationships with to do the NHP studies, then you will need to discuss with the CRO at that time. If you are asking NIH to conduct any of the CRO studies, you don't have to estimate the costs within your budget. You only have to include budget considerations for activities under which you will be doing in your own laboratory or through your own CRO relationships proposed.
Okay, that's all the questions we have, and we're almost out of time, so good timing.
Very good. Well, thank you, everyone. And as mentioned earlier, please contact Jules and myself. We look forward to hearing from you and some of the exciting research that you will be proposing under BPN-Biologics. See you next time, and look for follow-up emails with the PDFs and subsequent links to information following the webinar.